Okay, so we were done with the characteristics of enzyme. Now let's talk about the mechanism of enzyme action. It means that what are the things, what is the mechanism of enzyme action? So first of all, there is a substrate that bind with the active site to form an enzyme substrate complex. What does that mean? Means there is enzyme, and as you guys all know, particular enzyme is having a particular substrate, but at some places there is not, because just of the flexibility of the active site. So there's an enzyme, and if this is adding with a substrate is can be any kind of a substrate can be there. Okay, so it is making a enzyme substrate complex. When these both join together, we are calling as that as a enzyme substrate complex. After that, when enzyme substrate complex is making a product, okay, so product enzyme product is made. So after that, enzyme will got separate as well as product will got separate. Okay, so this product is finally released. Okay, so the product is attached with the, you can say that this one, a product is attached with the this one, so we can say that as an enzyme product complex. After that, enzyme will be detached and product will be formed. So basically enzyme is having a, for example, this is the active site of an enzyme. Okay, so this is what, this is the environment that is outside of the active site. So in active site, this active site, there should be some kind of a change. Maybe that can be the change of pH, that can be changes of water concentration. So this is the local condition inside the active site, which is different from the outside. So we are having two models of the enzymes of action. We are having two model. First one model is called the first one model is called a key lock model, lock and key model. What does it mean? It means that there is lock and key model, okay, which was described by a scientist, that is Emil Fisher. He said that there is, for example, there is any kind of a active site where substrate combined. He said this active site is rigid, which means it is having a rigid shape. So it means that it will bind with only specific substrate. Specific substrate will bind just because of the rigidness, specific substrate. Okay, so this is what, this is the, this enzyme is called the non-regulatory enzyme, which means the active site do not change because they are rigid. For example, we are having a, you can say that a enzyme that is sucrase work on glucose plus fructose. Okay, sucrase break down into the glucose into fructose, as well as maltase that break to glucose. Okay, so this is the enzyme, this enzyme will not work on that, this enzyme will not work on that. So this is, they are specific substrate with the specific enzyme. So this model is saying that, for example, we are having a key and lock model, which means that, as you guys all know, every lock is having a different key. So that you can say that a different key will not, we cannot use it through a different lock. So every lock is having a specific key. So that's why it is called the lock and key model, because of the, they are having a particular substrate. Okay, after that we are having a enzyme. For example, we are having an example, that is urease. Okay, so urea plus water is basically with the help of urease, the product is formed, that is ammonia plus carbon dioxide. So urease only work on urea, this is specific. So this specific substrate which work in a specific enzyme which work on a specific substrate, that is called a absolute specificity. Okay, so this was the first model. Next we are having that is the induced fit model. This was described by a Koshland. So this is basically opposite of the lock and key model. It means that in this model the active side is basically flexible. Which means that in this active side which is flexible, any substrate can be joined. In that one lock and key, they were the specific substrate with the specific enzyme, but here any, you can say, the substrate can be joined with this active side of the enzyme. So this was a point, and that is called a regulatory or allosteric, which means that it can join because of the flexibility. More than 100 substrate can be joined into active side, different. For example, I'm having a active side of an enzyme, that is this, and substrate is this. Okay, so this is like that. So this is becoming like that after bending this. So you can see that this is not a shape and this is not a, like, they are both are different. Okay, so we are having a active side, for example this. So after that we are having a this kind of a substrate that can be joined easily, can be joined into the key and lock model. But if we talk about this one, so you can see that these both are different but they are joining. So this is called the induced fit model, where the more than 100 substrate can be joined into a active site. Different substrate can be joined. Okay, for example, we are having a hemoglobin plus oxygen, and the enzyme that is work on that is a carbonic amylase, not amylase, anhydrase. This is an enzyme and this is making this is oxyhemoglobin product. So this is not only an enzyme that is work on this hemoglobin, this is an enzyme that work on many product. But as you guys all know, in urea, as I told you, so urea is only working on the urea, not any other product. Here you can see that this enzyme is working on another product, just because they are having a flexible active side. So this was the induced fit model. In the next video we'll talk about the energy of activation. This was the two model which are coming in the mechanism of the enzyme action.